A 16-year-old girl was going blind in my clinic, and we didn't have a clear diagnosis, we didn't know the right therapy, and we were unsure if there was a clinical trial that would help her. The reason was we didn't have a real-time view into the molecules and cells driving disease in her eye, and we couldn't biopsy the eye. Cell-level analysis in living humans is essential to understanding disease mechanisms, but biopsies of vital organs like the eye or brain are dangerous, if not impossible. Wouldn't it be amazing if in just a few drops of fluid, we could get the answers we needed?
It's possible. We developed a platform called Tempo. It allows us to trace thousands of proteins in liquid biopsies back to their cell origins. This protein cell map has the potential to significantly improve personalized patient care.
I'm Vineeth Mahajan. I'm a vitreoretinal surgeon, scientist, and professor here at Stanford University.
And I'm Julian Wolf, an ophthalmologist and postdoctoral fellow in the Mahajan lab.
There are 20 million cataract surgeries done worldwide each year, and in each case, a small amount of fluid is removed but thrown away. Here at the Byers Eye Institute, we collected eye fluid from 120 patients. The fluid was immediately frozen and carefully stored in a biobank.
From only a few drops of fluid from the front of the eye, we were able to measure approximately 6,000 different proteins using a DNA aptamer-based assay. That's a much higher resolution than previous measurements.
Measuring proteins in eye fluid is a technical challenge because of the small volume and also because the concentration of protein is 400 times lower than it is in blood.
We then took advantage of another technology called single-cell sequencing. It tells us which genes are turned on by single cells. From public databases, I collected data from over 80,000 cells from all 72 known cell types in the human eye. Then I looked at each of the nearly 6,000 proteins we found and mapped them back to the combined single-cell data. From this analysis, we know exactly which cell is producing which protein in the eye. We call this Tempo, tracing expression of multiple protein origins. Here is a human eye. You can see the retina in yellow. It's like a film covering the inside walls of the eye. The retina is made up of different cells that carry out specific functions. We could detect unique proteins released only by specific cells, shown here by the small circles.
When we zoom out again, we can see that the proteins are released into the vitreous, a watery gel that fills the inside of the eye, and these proteins migrate to the front of the eye where we can ID them. Using Tempo, we can perform cell-level analyses in living humans. It's amazing that without a tissue biopsy, we have molecular-level resolution where we can practically see inside living cells.
Blindness due to diabetes is increasing around the globe. Our liquid biopsies showed the main cells driving diabetic retinopathy changed from blood vessel cells in the early stage to immune cells in the late stage.
Parkinson's disease is a devastating disorder of the nervous system. We found that many brain proteins linked to Parkinson's can be measured in the eye. This is exciting because it means protein biomarkers in the eye could help diagnose Parkinson's or determine if a patient is responding to therapy.
We used artificial intelligence to predict the age of the eye based on protein patterns. Because we know the marker proteins of each cell, we could even develop AI clocks to predict the age of a specific cell type, like blood vessels, retinal cells, or immune cells. We found that the eye is about 30 years older in patients with late-stage diabetic retinopathy compared to healthy humans. In many patients with diabetes, there are no visible symptoms of eye disease. We found that in those eyes, the age of the immune cells and of retinal cells is about 10 years older than healthy eyes. It means there is accelerated aging even before clinical signs of disease can be seen by an ophthalmologist.
Even eyes that had been treated successfully, where the bleeding had stopped or the inflammation was controlled, showed accelerated aging. So, anti-aging therapies may be needed to fully reverse damage from disease.
In principle, Tempo can be applied to other organ systems, such as the brain, lung, or kidney.
We believe Tempo and our AI approach fill a critical gap in our ability to study disease in living humans.